आई विश कि हम दोनों की स्कोर ना एक्सचेंज हो जाए अयान, फिर तुम बोलते। हाँ निधि, बहुत पढ़ लिया होगा ना, मैंने तो टाइम वेस्ट ही किया इतने टाइम तक। तुम तो कब से पढ़ रही होगी? आई थिंक 8 से ना निधि। सर्वजिता पे वीडियो लेक्चर सब कुछ देख रही थी, वो लेक्चर नहीं देख रही थी शायद, रिवीजन सीरीज कुछ देख रही थी। कॉन्स्टिपेशन भी फेस किया होगा। निशान कहाँ गए थे पहले? इससे पहले भी मैं आई थी। मैंने क्वेश्चन करने की ट्राई कर रही थी। ओह क्वेश्चन, ओके ओके। अगर ना नींद अच्छे से लिया हो ना तो फिर पढ़ने का भी दिल करता है, नींद अच्छे से नहीं लिया है तो पढ़ने का दिल नहीं करता। आई थिंक शुभम आप फिर से सोने गया है ना। एक आता है दूसरा चला जाता है। चलो स्टार्ट ही करते हैं, क्या ही कर सकते हैं। आई एंड मोटा। हाँ तुम्हें लगा होगा, लेकिन मुझे तो मोटा ही लगा, मोटे इंसान। हेलो मिस, आई एम न्यू हियर। हाय श्रेयस। तुमने सो लिया क्या ब्रेक में? हाँ सोने गई तो देखो ना, ब्लैंकेट पे खोल के उधर फेंक दिया है, लेकिन नींद ही नहीं आई तो मैं वापस से आ गई। मर्फो हो गया क्या इमा? किस चीज़ की मर्फो? मॉर्फोलॉजी ऑफ फ्लावरिंग प्लांट 3 बार कर लिया है ऐसा लगा है यहाँ पर। मुझे नहीं पता, लेकिन तुम क्वेश्चन पूछोगी तो मुझे शायद याद नहीं होगा। लेकिन यहाँ पर तो 3-4 बार किया है। 1 2 3 4, 4 राइट लगाया है तो 4 बार किया है। प्लांट किंगडम ना मेरे 6 टाइम्स किया है, लेकिन अभी पूछोगी तो मुझे कुछ नहीं आता। मैंने सबसे कम जो किया है ये एवोल्यूशन है भाई साहब। एग्जैक्टली। 40 मिनट का सेशन रखा है। ओके, ज़्यादा लंबा नहीं रख सकती क्योंकि फिर मेरा पढ़ने का दिल ही नहीं करेगा, इसलिए छोटा छोटा सेशन में कंप्लीट करेंगे। ओके ना, माई एग्जाम इज क्रैकिंग मी। अगर मैं क्रैक करती तो अच्छा होता ना, इसलिए तो आयरन ही लिखा है। Okay, good luck Nish, good luck निधि एंड गुड लक अयान. आई थिंक एस बी नहीं आएगा आज. मैं चैट्स ऑफ कर दूंगी, ब्रेक में कर लूँगी. एग्ज़ाम से बेटर लक नेक्स्ट टाइम. एग्जैक्टली आयुष, मेरे साथ तो यही हो रहा है, दूसरों के साथ आई डोंट नो. गुड लक अयान, गुड लक एवरीवन. 40 मिनट का लगाया है. ओके, ब्रेक में वी विल टॉक. स्टार्ट कर रही मैं टाइमर. ओके, पागल हो जाएंगे पढ़ते पढ़ते, आपको भी दिमाग में जाए तो एक बात ना. Have you ever even wondered why an elephant always give birth only to a baby elephant and not some other animals, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These are several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parents to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Variation is a degree by which progeny differ from their parents. Human knew from as early as 8,000 to 8,000 to 8,000 to 1,000 BC that one of the cause of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed desirable character. For example, through artificial selection and domestication from ancestral wild cow. For example, through artificial selection and domestication from ancestral wild cow. We all know Indian breeds. Example, Shahiwal cows in Punjab. Well known Indian breeds. Example, Shahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of character and variation, they had very little idea about the scientific basis of this phenomena. Okay. Mendel law of inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, 1856 to 1863. 1856 to 1863. 1856 to 1863. 1856 to 1863 and proposed the law of inheritance in living organism and proposed the law of inheritance by Gregor Mendel, 1856 to 1863. Gregor Mendel conducted hybridization experiment on garden peas for 7 years, 1856 to 1863. 1856 to 1863, garden peas. Gregor Mendel gave law of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiment had a large sampling size. Which gave greater credibility to the data that he collected. Large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generation of his test plants proved that his results pointed to general rules of inheritance. Also, the confirmation of his inferences from experiments on successive generation of his test plants proved that his results pointed to general rule of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plants, yellow or green seed. This allowed him to set up a basic framework of rules governing inheritance. Which was expanded on by later scientists to account for all the diverse natural observations and the complexities inherent in them. Mendel conducted such artificial pollination cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination shows the stable trait inheritance and expression for several generations. Mendel conducted such artificial cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and express for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except one character with contrasting trait. Mendel selected 14 true breeding pea plant. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character. With contrasting traits. Some of the contrasting traits selected were smoother wrinkled seeds, yellow or green seeds, inflated or constricted green or yellow pods, full or constricted green or yellow pods, and tall or dwarf plant. Figure 4.1 and table 4.1. 7 pairs of contrasting traits in pea plants studied by Mendel. Seed color, seed shape, then flower position, flower color, stem height, pod shape and pod color. Seed shape, dominant and recessive. Seed shape, dominant, round and wrinkled. Seed shape, round and wrinkled. Seed color, yellow and green. Flower color, violet and white. Pod shape, full constricted, inflated and constricted. Pod color, green and yellow. Violet, dominant, full, dominant. Green color, dominant. Green color of pod, dominant. Axial position of flower, dominant. Terminal position of flower, recessive. Stem height tall and dwarf. Okay, yellow green seed color. 7 pair of contrasting trait in pea plant studied by Mendel. 7 pairs of contrasting trait. 7 pairs of contrasting trait studied by Mendel. 7 pairs of contrasting trait in pea plant studied by Mendel. 7 pairs of contrasting traits in pea plant studied by Mendel. 7 pair of contrasting trait. 7 pairs of contrasting trait. 7 pairs of contrasting trait in pea plant studied by Mendel. 14 true breeding pea plant. 14 true breeding pea plant contrasting traits studied by Mendel in pea. Stem height. Paisam satayum. Stem height tall, dwarf, flower color violet and white, flower position axial and terminal, pod shape inflated and constricted, pod color green and yellow, seed shape round and wrinkled, seed color yellow green. Okay. Contrasting trait studied by Mendel in P. Contrasting traits studied by Mendel in P. Characters. Stem height, tall and dwarf. Contrasting trait, tall and dwarf. Flower color, violet and white. Flower position, axial and terminal. Pod shape, inflated and constricted. Inflated and constricted. Inflated and constricted. Okay. Axial and terminal. Axial and terminal. Violet and white. Flower color, stem height, tall and dwarf, pod color green and yellow, seed shape round wrinkled, seed color yellow green. Inheritance of one gene. Inheritance of one gene. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study the inheritance of one gene. To study the inheritance of one gene. Mendel crossed tall and dwarf pea plant. He collected the seed produced as a result of this cross. He collected the seeds produced as a result of this cross, and grew them to generate plant of the first hybrid generation. This generation is also called the filial progeny, or F1 progeny. F1. Mendel observed that all the F1 progeny plants were tall, like one of its parent, none were dwarf. He made similar observation for the other pair of traits. He found that the F1 always resembled either one of the parents, and the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants, and to his surprise found that in the filial 2 generation, some of the offspring were dwarf. The character that was not seen in F1 generation was now expressed. The proportion of plants that were dwarf were one-fourth of the F2 plants, while 3/4 of the F2 plants were tall. 3/4 were tall, only 1/4 were short. Okay. Mendel then self-pollinated the tall F1 plants, and to his surprise found that in the filial 2 generation some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, where 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending. Okay. That is, all the offspring were either tall or dwarf. None were of in between height. Similar results were obtained with the other traits that he studied. Only one of the parental trait was expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion of 3:1. The contrasting trait did not show any blending at either F1 or F2 stage. Petal, stigma, anther, stamen, Carpel, petal, stigma, anther, stem, carpel. Removal of anther, emasculation. Parental, transfer of pollen. Removal of anther, emasculation. Female parent, male parent with a transfer of pollen, pollination. Steps in making of cross and pea. Okay, everything is told. All the plants were told. Based on the observation, Mendel proposed that something was being stably passed down. Based on this observation, Mendel proposed that something was being stably passed down unchanged from parents to offspring. Passed down unchanged from parents to offspring. Based on this observation, Mendel proposed that something was being stably passed down unchanged from parents to offspring through the gamete over successive generations. He called these things as factors. Now we call them as genes. Factor and genes are similar. Factors and genes are similar. Okay, factors and genes are similar. It's already 11 o'clock. Factors and genes are similar. He called these things as factor. Now we call them as genes. Genes therefore are the unit of inheritance. Genes therefore are the unit of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different form of the same gene. Genes therefore are called unit of inheritance. So genes, unit of inheritance, also known as factor in the time of Mendel. At the time of Mendel, they contain the information that is required to express a particular trait in an organism. They contain the information that is required to express a particular trait in an organism. Genes which code for pair of contrasting trait, example known as allele. Genes that code for a pair of contrasting trait are known as alleles. That is, they are slightly different form of same gene. Alleles are the slightly different form of same gene. Alleles are the slightly different form of same gene. Alleles are the slightly different forms of the same gene. Alleles are the slightly different forms of the same gene. If we use alphabetical symbols for each gene, okay, and let's read it again. Gene contains the information that is required to express a particular trait in an organism. Gene possess. Is it the word possess? Yeah, possess or contain the information that is required to express in the particular trait. That is required to express a particular trait in an organism. That is required to express a particular trait in an organism. That is required to express a trait in an organism. Particular trait in an organism. Genes which code for a pair of contrasting traits. Gene which is responsible for gene which codes for a pair of contrasting trait are known as alleles. They are slightly different forms of same gene. Alleles are slightly different forms of same gene. If we use alphabetic symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of character of height, T is used for tall trait and small t for the dwarf. Capital T and small t are alleles of each other. These two, capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be Tt, capital T small t, or small t small t. Mendel also proposed that in a true breeding, tall or dwarf P variety, the allelic pair of gene for height are identical or homozygous, capital T and small t respectively. Capital T and small t are called the genotype of the plant, while this respective terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T and small t? As Mendel found the phenotype of the F1 heterozygote capital T and small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, as in the F1. One dominates the other, as in the F1, and hence is called a dominant factor, while the other factor is recessive. In case of capital T for tallness is dominant over small t for dwarfness, that is recessive. Okay, small t is recessive. Okay, dwarfness. He observed identical behavior for all the other characters traits pairs that he studied. He observed identical behavior for all the other character or traits pair that he studied. He observed identical behavior for all the other characters trait pairs that he studied. It is convenient and logical to use the capital and lowercase of an alphabet symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small t for dwarf, because you will find it difficult to remember whether capital T and small t are alleles of the same gene, character or not. Alleles can be similar as in case of homozygotes, capital T and small t, or can be dissimilar in case of heterozygotes, capital T, small t. Since the capital T small t plant is heterozygous for genes controlling one character height, it is monohybrid, and the cross between capital T and small t is a monohybrid cross. Monohybrid and monohybrid. It is monohybrid. Okay. Plant से monohybrid, cross से ये monohybrid cross. Okay. Parental capital. Tall and dwarf. F1 generation tall selfing. 3:1 ratio. Tall and dwarf. Okay. F2 generation. Diagrammatic representation of monohybrid cross. Diagrammatic representation of monohybrid cross. Capital T small t plant is heterozygous for genes controlling one character height. Capital T small t plant is heterozygous. Capital T small t plant is heterozygous. The capital T small t plant is heterozygous for genes controlling one character height. It is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending. From the observation that the recessive recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and the dwarf plant produce gamete by the process of meiosis, the alleles of the parental pair separate or segregate. the process of meiosis, by the process of meiosis. When the tall and the dwarf plant produce gamete. When the tall and the dwarf plant produce gamete. When the tall and the dwarf plant produce gamete. When the tall and the dwarf plant produce gamete by the process of meiosis, the allele of the parental pair. The allele of the parental pair separates or segregates from each other, and only one allele is transmitted to a gamete. When the tall and dwarf plant produce gamete. When the tall and dwarf plant produce gamete. Tall र dwarf plant gamete produce गर्छ त्यतिखेर हुन्छ by the process of meiosis, the allele of the parental pair, the alleles of the parental pair separate or segregate from each other. The alleles of the parental pair separate or segregate from each other. The alleles of the parental pair separates or segregate from each other. When the tall and dwarf plant produce gamete by the process of meiosis, tall and dwarf plant produce gamete by the process of meiosis, the allele of the parental pair separate or segregate. The allele of the parental pair separate or segregate from each other, and only one allele is transmitted to the gamete. The only one allele is transmitted to the gamete. The allele from the parent segregate or separate. The allele of the parental pair. The allele of the parental pair segregate or separate. The alleles of the parental pair. The alleles of the parental pair separate or segregate from each other, and only one allele is transmitted to the gamete. When the tall and the dwarf plant produce gamete by the process of meiosis, all the allele of the parental pair separate or segregate from each other, and only the allele is transmitted to the gamete. Only one allele is transmitted to the When the tall and the dwarf plant produce gamete by the process of meiosis, the allele of the parental pair separate or segregate from each other, and only one allele is transmitted to a gamete. This segregation of alleles is random. This segregation of allele is a random process, and so there is 50% of chance of a gamete containing either allele, as has been verified by the result of the crossing. In this way, the gamete of the tall plant, capital T, have the allele capital T, and the gamete of the dwarf, small t plant, have the allele small t. During fertilization, the two alleles, capital T from one parent C through the pollen and small t from the other parent, then through the egg, are united to produce zygote that have one capital T and one small t allele. In other words, the hybrids have capital T and small t. Since these hybrids contain alleles which express contrasting traits, the, the plant are heterozygous. Since these hybrids contain alleles which express contrasting traits, since these hybrids containing alleles which express contrasting traits, the plants are heterozygous. The plants are heterozygous. These hybrids contain alleles which express contrasting traits. The plants are heterozygous. Contrasting traits. The plants are heterozygous. The production of gamete by the parents, the formation of zygotes, the F1 and F2 plants can be understood from the diagram called Punnett square. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parent, formation of the zygote, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in figure 4.4. It was developed by a British geneticist, Reynold C. Punnett. Okay, British geneticist, Reynold C. Punnett. It was a graphical representation to calculate the probability of all possible genotype of offspring. Calculate the probability of all possible genotype of offspring in the genetic cross. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented. All possible combinations are represented in box below in the square, which generate a square output form. A Punnett square used to understand a typical monohybrid. A Punnett square used to understand a typical monohybrid cross conducted by Mendel. A Punnett square used to understand typical monohybrid cross conducted by Mendel. B. Between true breeding tall plant and true breeding dwarf plant. True breeding timber. True breeding tall and dwarf plant. Okay. Tall and dwarf plant. True breeding do they. Capital T, small t. F1 generation. Capital T, small t. 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 Small t capital small. Capital small. F1 generation all tall. F1 TT. Capital T. Capital T, small t. capital T, small t, small t, small t. Phenotypic ratio 3:1. Genotypic ratio 1:2:1. Genotypic ratio in F2 generation 1:2:1 in monohybrid cross. Monohybrid cross monohybrid cross 1:2:1. Genotypic ratio, phenotypic ratio 3:1. 1:2:1. Genotypic ratio, phenotypic ratio 3:1. So before I go, मच्छर के बच्चे, तो नहीं क्या करूँगी मैं एग्ज़ाम में जाके, कुछ तो करेगी ही. Boxes below in the square which generates a square output form. The Punnett square shows the parental capital. Okay. The Punnett square shows the parental tall TT male and dwarf small t plants, the gamete produced by them, and the F1 capital T small t progeny. The F1 plant of genotype capital T and small t are self-pollinated. The symbol positive, okay, this one is for female and this one is for male, denotes the female egg and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t when self-pollinated produce gamete of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grain of genotype capital T have 50% chances to pollinate egg of the genotype capital T as well as of genotype small t. Punnett square shows the parental capital. Okay. Punnett square shows the parental tall capital T male and dwarf small t female plant, the gamete produced by them, and F1 capital T small t progeny. The F1. Punnett square shows the parental tall capital T male and dwarf small t female plant, the gametes produced by them, and the F1 capital T small t progeny. Punnett square. Capital T male and dwarf small t female plant, the gamete produced by them, and the F1 capital T small t. I think that will be fine. Self-pollinated, the symbol denote the female egg and male pollen. Self-pollinated produce gamete of genotype capital T and small t. मुझे एग्जाम देना ही नहीं है. In equal proportion. When fertilization takes place, the pollen grain of genotype capital T have a 50% chance to pollinate egg of the genotype capital T have a 50% chance to pollinate egg of genotype capital T as well as of genotype small t. Also pollen grains of genotype small t have a 50% chance. 50% chance. Capital T and F1 have the genotype capital T small t. When self-pollinated produce a gamete capital T and small t in equal proportion. Okay. When fertilization takes place, the pollen grain of genotype capital T chance to pollinate egg of genotype capital T as well as genotype small t. As a result of random fertilization, the resultant zygote can be of the genotype capital T, small t, or small t, small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization leads to capital T, half 1/2 leads to capital T small t, and 1/4 of small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall at F2. 3/4 of the plant are tall, where some of them are capital T, while others are capital T and small t. Eternally, it is not possible to distinguish between the plant with the genotype capital T and capital T and capital T small t. Hence, when the genotypic pair capital T small t, only one character tall is expressed. Within the genotype capital T small t, only one character tall is expressed. Hence, the character capital T or tall is said to dominate over the other allele small t or dwarf character. It is thus due to the dominance of one character over the other that all the F1 are tall, though the genotype is capital T small t. And the F2, 3/4 of the plant are tall, though genotypically 1/2 are capital T small t, and only 1/4 of are capital T of the plants, okay, are tall. 3/4 of the plants are tall. Heterozygous and one is homozygous. This led to the phenotypic ratio 3/4 tall, 1/4 capital T, 1/2 capital T small t, and 1/4 small t in the ratio 3:1, but a genotypic ratio of 1:2:1. Okay. कितने मिनट का आप ब्रेक ले? स्लॉट कंप्लीट हो गया तो break. Those who want to take break, they can take. आधे टाइम सोचते ही चला गया, क्या सोचे इतना ज़्यादा? मैं तो कुछ ही नहीं सोची। यार मुझे बहुत डर लग रहा है, मुझे एग्ज़ाम देने का भी दिल नहीं कर रहा है। आज शुभम नहीं है, वरना वो हमेशा एक डॉट छोड़ देता था जल्दी जल्दी। May 4 शाम को कंसर्ट है, उसमें सोनू निगम आने वाला है। वो ये सोच रही थी, यार तुम लोगों का कितना सही है। या तुम्हें तो खुश होना चाहिए, तुम कंसर्ट में जा रही हो। सोने गया ऐसा लग रहा है, मुझे भी नहीं पता है, एलेक्स को ही पता होता है इसके बारे में तो। हाँ ये भी है, पढ़ते रहना चाहिए। चूहा ऊपर कुछ तो खा रहा है, पेपर खा रहा है शायद। वहाँ पर पेपर रखा था शायद सीलिंग में, वही खा रहा है, आवाज आ रहा है। यार निधि मैं सोने जाऊँ क्या? इतनी मुश्किल से पढ़ने आती हूँ, फिर सोने की ही बात करती रहती हूँ। नहीं, तुमसे पूछ रही हूँ। हाई स्टडी विद सोमिया। क्यों पूछा? शायद अनोइंग। नील डाउन। 2026 भी लग ही जाएगा। सोमिया। मुझे तो नहीं पता, व्हाट डू यू मीन किस प्लेटफॉर्म से प्रिपेयर कर रहे हैं? नीट के दूसरे दिन ही ईमा। नहीं, तुमने ना अच्छे से प्रिपेयर किया होगा तो तुम्हारा इस बार में ही हो जाएगा ना, तो ड्रॉप लेने की क्या ज़रूरत है? बाय अयान। आई भाइयों पर की और सिलेक्शन भाई साहब, सुन के ही अजीब लग रहा है। निधि स्टार्ट करे एक और स्लॉट। निधि what she is giving me attendances also. एक्टिंग मत कर सासके, तू अपना पर मैं अपना पढ़ती हूँ, देखते हैं कौन जीतेगा। आर्टिफैक्ट्स, अरे सेट पर भी ध्यान दे तू। हड्डियाँ दिख रही है, ये तो अम्मा की लाइन चिपका दिया है इसने। आपकी सीट खा जाऊंगा। लगता है सीट में हक देना पड़ेगा, फिर ये गू भी खाएगा सीट के साथ। मैं ना यानी के साथ रह रह के उसकी उसकी खूबी मेरे में आ गई है शायद। ओके मैंने चैट ऑफ कर दिया। ओके हाय आयशा। अब नेक्स्ट वीक में ये चैट ऑन होगा। ओके मुझे ना ये सासके के लिए बहुत बुरा फील हो रहा है। इट्स ओके है ना, मेरा वो मतलब नहीं था कि सासके। क्लिप तो मैं स्टार्ट कर रही हूँ। ओके टाइमर। आई थिंक सासके चला गया है, लेकिन क्या ही कर सकते हैं। मैं इतना हंसी कंट्रोल करने की वो कोशिश कर रही हूँ। मुझे सोच सोच के हंसी आ रही है। मुझे सोच सोच के हंसी आ रही है वो बात पर, पता नहीं क्यों। मैं टाइमर स्टार्ट कर रही, अब मत सोचूं, अब नहीं सोचने की कोशिश करूंगी। ओके, गुड लक एवरीवन। 1/4, 1/2 and 1/4 ratio. Capital T, capital T, capital T, small t, small t, small t is a mathematically condensable to form of the binomial expression ax plus by whole square that has the gamete bearing genes capital T or small t in equal frequency. The expression is expanded as given below. 1/2 t plus 1 by t, half t plus half t square, small t whole square, a plus b, a square plus b square, a square plus b square plus 2ab. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generation. He concluded that the genotype of the dwarfs was homozygous, small t, small t. What do you think he would have got had he self-pollinated a tall F2 plant? Mendel self-pollinated the F2 plant and found that F4 generation, F3 and F4 generation. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarfs was homozygous, small t, small t. What do you think he would have got had he self-pollinated a tall F2 plant? Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous, small t, small t. He concluded that the genotype of the dwarfs was homozygous. He concluded that the genotype of the dwarfs was homozygous, small t, small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotype composition. That is, for example, whether a tall plant F1 or F2 has capital T capital T or capital T small t composition. Tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant. To determine the genotype of tall plant at F2, Mendel crossed the tall plant. Mendel crossed the tall plant from F2 with the dwarf plant. मुझे फिर से वो सास्के का याद आया. भाई साहब सास्के फिर he is my Yanni. Test cross to determine the genotype of a tall plant. 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 To determine the genotype of a tall plant in F2 generation. Test cross words, okay? To determine the genotype of a tall plant in F2 generation, Mendel crossed the tall plant with the recessive female parent. Recessive F2. With the dwarf parent, okay? Dwarf plant. This he crossed a test. This he called a test cross. Like eventually test cross भन्यो. Test cross मा के गर्छ? Recessive parent सँग recessive parent dwarf plant सँग के गर्छ? F2 generation लाई cross गर्छ. In a typical test cross, an organism P plant here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5, which is 4.5, shows the result of typical test cross where violet color flower W is dominant over white color flower small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using Punnett square, try to find out the nature of the offspring. Using Punnett square, try to find out the nature of the offspring of a test cross. What ratio did you get using the genotype of this cross? Can you give a general definition for a test cross based on his observation? Okay, छोड़ दें यहाँ सब diagrammatic representation of a test cross. Recessive female parent. Recessive parent female. Homozygous and heterozygous. Homozygous recessive, heterozygous recessive. Homozygous recessive and heterozygous recessive. यस्तो हुन्छ त नि homozygous recessive मा यो पनि homozygous हेट्रो हैन यो पनि homozygous recessive This is dominant phenotype, WW. All flowers are violet. Unknown flower is homozygous. Unknown flower is homozygous dominant. Homozygous dominant result. Interpretation. All flowers are violet. Interpretation. Unknown flower is homozygous dominant. Heterozygous. Okay, dominant phenotype. Genotype unknown. Heterozygous. WW. 50%, 50% violet, 50% white, 50% violet, 50% white, 50% violet, 50% white. Half of the flower are violet and half of the flower are white. Unknown flower is heterozygous. Unknown flower is heterozygous. Unknown flower heterozygous छ भनेर कति बेला थाहा पाउँछ भन्दाखेरि 50% violet कलर आउँछ 50% मा कुनै कलरै आउँदैन. Half of the flower are violet and half of the flower are white. Half of the flower are violet and half of the flower are white. Half of the flowers are violet and half of the flowers are white. Unknown flower is heterozygous. If unknown flower is heterozygous, half of the flower are violet and half of the flower are white. If unknown flower is homozygous dominant, then all the flowers will be violet. All the flowers will be violet. Okay. Pouring mercury. Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called principle of law of inheritance. The first law of मुझे ना एग्जाम देने जाने का दिल नहीं कर रहा है. Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principle or law of inheritance, the first law or law of dominance, and second law or law of segregation, law of dominance. Characters are controlled by discrete units called factors. Law of dominance. Characters are controlled by discrete unit called factors. Factors occur in pair. In a dissimilar pair of factor, one member of the pair dominates the other recessive. Okay, law of dominance. Factors occur in pair. Characters are controlled by the discrete unit called factors. Characters are controlled by discrete unit called factors. Factors occurs in pair. In a dissimilar pair of factor, one is completely dominant. One member of the pair dominant, the other recessive. एउटाले dominate गर्छ अर्को recessive लाई के यही तिनटा कन्क्लुजन देख्थ्यो. Factors, charactersहरूलाई केले कन्ट्रोल गर्छ? Factorsले कन्ट्रोल गर्छ. Characters are controlled by discrete unit called factors. Factors occur in pair. In dissimilar pair of factor, one member of the pair dominates the other recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Weird लग रहा है ना, लेकिन कोई बात नहीं, सही तो है. Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called principle of law of inheritance. Principle or law of inheritance. Principle of inheritance or law of inheritance. The first law of dominance, and the second law of segregation. First law of dominance. Second law of segregation. Law of dominance, law of segregation. Law of dominance. Characters are controlled by a discrete unit called factors. factors occurs in pairs. Characters are controlled by a discrete unit called factors. Factors occurs in pair. In a dissimilar pair of factor, one is dominant over the other, recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1. Okay, is used to explain the expression of only one of the parental character. Expression of only one of the parental character in the F1 generation of monohybrid cross of P cross. The expression of only one of the parental character in monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 3:1. It also explains the proportion of 3:1 found at F2. The law of dominance is used to explain expression of only one of the parental character in a monohybrid cross in the F1. The law of dominance is used to explain the expression of only one of the parental character in monohybrid cross. The expression of only one of the parental character in monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at F2. It also explains the proportion of 3:1 explained at F2. It also explains the proportion of 3:1 obtained at the F2. 3:1 obtained at the F2. It also explains the proportion of 3:1 obtained at the F2. 3:1 obtained at the F2. Obtained at the F2. 3:1 obtained at the F2. Characters are controlled by discrete unit called factors. Factors occurs in pair. In a dissimilar pair of factor, one member of the pair dominates the other recessive. The law of dominance is used to explain the expression of only one parental character in monohybrid cross in F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Law of segregation, another second law. Okay, first law of dominance, second law of segregation, and the third law में तो भी पहुंचे ही नहीं है. Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation. Law of segregation. This law is based on the fact that the allele do not show any blending and that law is based on the fact that alleles do not show any blending and that both the character are recovered and that both the character are recovered as such in F2 generation, though one of this is not seen at the F1 generation. This law of segregation is based on the fact that alleles do not show any blending. Alleles do not show any blending. Alleles do not show any blending, and that both the characters are recovered as such in F2 generation. And both the characters are recovered as such in the F2 generation, though one of this is not seen at the F1 stage. Though the parent contained two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors from each other, such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Allele do not show any blending allele do not show any blending. Allele do not show any blending. 12. Okay. Law of segregation. The law is based on the fact that the allele do not show any blending and that both the characters are recovered as such in the F2 generation. Though one of these is not seen at the F1 stage, though the parent contains two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gamete, each having one allele with equal proportion. The characters are recorded as such in F2 generation. Do not show any blending, so the characters are recorded as such in F2 generation. Though one of this is not seen at the F1 stage. Though one of this is not seen at the F1 stage. Though one of this is not seen at the F1 stage. Though the parent contains two alleles during gamete formation. Though the parent contains two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Though the parent contains two alleles during gamete formation, the factors or alleles of one pair segregates from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Each having one allele with equal proportion. F2 generation but recovered also, though one of these is not seen at the F1 stage. So the parent contains two alleles during gamete formation. The factor or alleles of a pair segregate from each other such that the gamete receives only one of the two factors. Though the parent contains two alleles during gamete formation. Contain two alleles during gamete formation. During gamete formation, the factors or allele of a pair segregate from each other such that gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gamete, each having one allele with equal proportion. Heterozygous one. Incomplete dominance and codominance are the exception of law of dominance. Incomplete dominance. When experiments on bees were repeated using other traits in other plants, it was found that sometimes the F1 had phenotype that did not resemble either of the parent and was in between the two. The inheritance of flower color in the dog flower, snapdragon, or antirrhinum sp is a good example. Inheritance. Okay, dog flower, snapdragon, or antirrhinum is a good example. To understand incomplete dominance in a cross between true breeding red flower RR and a true breeding white flower small r, that F1 capital R small r was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: capital RR, following ratio. Okay, 1 capital R red, 2 capital R small r pink, and 1 small r white. There here are the phenotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratio had changed from 3:1 dominant to recessive ratio. Was that R what happened was that R was not completely dominant over small r, and this made it possible. What happened was that capital R was What happened was the capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from RR red to small r white. Explanation of concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must know that what a gene does. Every gene, as you know by now, contain the information to express a particular gene. Okay, every gene contains the information. Every gene contains the information to express a particular trait. In a diploid. Okay, every gene contains the information to express a particular trait. Every gene contains a particular trait to express in a contain a particular information. Every gene contains a particular information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of allele. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical. There are two copies of each gene, that is, a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One human may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now, there are two copies of this gene, the two allelic form. Let us assume, as is more common, that the normal allele produce the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal, less efficient enzyme, a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to the non-modified allele. Modified allele, okay? Equivalent to the non-modified allele, that is, it will produce the same phenotype trait, that is, result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produce a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally recessive allele. Original phenotype, dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is due to non-functional enzyme. Recessive trait is due to the non-functional enzyme or because no enzyme is produced. No enzyme is produced or non-functional enzyme. No enzyme or non-functional enzyme. हिंदी आ रही है तो मुझे हिंदुली In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype trait, that is, result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produce a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning. ये वाला थोड़ा मुझे और एक बार करना चाहिए ना. बाकी तो कर ही लेंगे, इनहेरिटेंस जिम भी कर लेंगे. पॉलीजेनिक इनहेरिटेंस प्लियोट्रोपिया लगी सही तो दिया है. In case and recombination, sex determination दिया है. Pleiotropial polygenic inheritance. Oh, it's given. Polygenic inheritance. Light skin color, human skin color, right? Sex determination in human, sex determination in honeybee. और इसमें दिया था, मैं ही पागल लड़की पुरानी बुक फॉलो कर रही थी. म्यूटेशन, पेडिग्री एनालिसिस, कलर ब्लाइंडनेस भी दिया था, मुझे पता ही नहीं था. Pedigree analysis, Mendelian disorder, hemophilia, sickle cell anemia, phenylketonuria एंड थैलेसीमिया भी तो दिया है इतना ज़्यादा. क्रोमोसोमल डिसऑर्डर उसके बाद डाउन सिंड्रोम की बात हुए. मैं कहाँ पर थी, मैंने ठीक से ये ना सेशन कंप्लीट भी नहीं किया. ये वाला मैं अगेन करूँगी ऐसा बोल रही थी ना. चलो मैं ये ऑन कर देती हूँ फिर से, एक और बार ऑन कर देती हूँ. हालांकि मेरा तो ब्रेक ही ब्रेक है, होना तो नहीं चाहिए ये वाला ब्रेक, लेकिन फिर भी कर दिया. Nidhi, are you there? Nidhi, are you there? Nishant, are you there? ओके ओके। शुभम कहाँ गए थे? हम लोगों ने तुम्हें मरा हुआ घोषित कर दिया था। भाई मैं चला सोने। हाँ अभी तक हो। मुझे लगा कि मैंने तुम्हें बोला ना कि 30 वाला, तो तुम गुस्सा होके चले गए। सास के समोसे वाला। I don't need nobody. I don't need nobody but you. कहाँ कहाँ ढूंढा तुझे, ना ना मुझे लिरिक्स ही नहीं पता, फिर भी मैं कोशिश तो करती हूँ ना गाने की। ये मेरे लिए बड़ी बात है। I don't need nobody but you. Good night, Gambusha. Good night. और हड़े मंदिर ना गुड नाइट। क्यों आया था पूछा नहीं नींद को। ये तुम सो गए। तुम मुझे समझ में ही नहीं आया। मैंने रीड तो कर लिया। अब गिल्टी हो रहा है। मेरा 7 दिन बाद एग्जाम है और मुझे ठीक से कुछ याद नहीं। मुझे होना चाहिए ना गिल्टी तो, कि पता नहीं क्या पढ़ा, रिवीजन भी ठीक से नहीं किया, प्लानिंग भी नहीं किया। ऐसा लग रहा है सब कुछ बर्बाद मैंने ही किया। मेरा नेट शायद खराब होने वाला है। पढ़ने वाली बात, खूब सारा पढ़ने वाली बात। अंतराक्षी। मैं जाके कुछ खा लेती हूँ ना। पता है इसका ना, ये नोज पैड लूज हो गया है और ये हमेशा ऐसा होता रहता है। फिर ऐसे मैं इसको पुश करती हूँ, फिर ऐसे हो जाता है। आई थिंक इसका ना स्क्रू टाइट करना पड़ेगा समथिंग, मुझे नहीं पता। कुछ तो करना पड़ेगा इसका। हाय राज। Yeah, it is. ये यूट्यूब वाले स्टिकर भैया को नहीं पता है कि मेरा चैनल है। मुझे पता है कि भैया का चैनल है, लेकिन ये मैंने खुद बनाया है। मैं उनकी इतनी बुराई करती रहती हूँ। अगर उन्हें पता होता तो मैं बिल्कुल बुराई नहीं करती उनका। मुझे ये वाला पहले कैट वाला पसंद था, अभी ये वाला मोदी जी वाला पसंद है। मैं एग्ज़ाम बाद यानी से पूछ के खूब सारा अपलोड करूँगी, लेकिन आई डोंट थिंक वो परमिशन देगा तब तक। अगर मैं 500 लेके आती तो शायद दे देता। मैंने बोला ना कि मुझे यूट्यूब करने दो, प्लीज़ प्लीज़ प्लीज़, मैं 500 से स्कोर करूँगी। फिर उसने बोला कि हाँ ठीक है, अच्छे से पढ़ना एकदम अच्छे से। फिर मैंने अच्छे से ही पढ़ा था, रिवाइज ही नहीं किया। भाई मुझे इतना बुरा लगता है ना मेरे दोस्त लोगों के लिए, वो हमेशा मेरे लिए प्रमोट करते रहते हैं। और निधि तो बेचारी, जब हम लोग बहुत पहले की बात है, बहुत पहले की बात है, जब सिर्फ हम लोग फ्रेंड्स ही होते थे ना, तब से वो ऐसा बोलती रहती है। थैंक यू। हाँ निधि, कोई बात नहीं है राज, हम सभी सेम लाइन में हैं, तभी तो पढ़ाई कर रहे हैं। मैं चाय बना के आती हूँ। तुम लोग स्टार्ट भी करने लगे हो, सब लोग गुड लक बोल रहे हैं। मैं डॉट 2 बजे पीऊँगा, मैं डॉट 2 बजे पीऊँगा। वन समथिंग तो हो ही तो गया है। पता नहीं क्या हो रहा है, ये क्या हो रहा है। टेस्ट है क्या कल? यस। एक कॉफी प्लीज। ये स्टिकर ये भी था, मुझे नहीं पता था। लेट मी चेक। मेरे पास नहीं है इतना क्यूट स्टिकर। को नहीं है तो यहाँ पर उसके बाद उसने कुछ बोला ही नहीं। नाइस नाइस। Kiba kamu? Good luck Nidhi. I'll try. इतने सारे मच्छर आ गए ना, मैं क्या करूं। मैं चैट ऑफ कर रही हूँ के। ये कौन है जो ऐसा बोल रहा है? थैंक यू नीट 2026 वॉरियर। हर रोज आके तुम मुझे ऑल द बेस्ट बोलते हो। ऐसा लग रहा है कि मैं जंग में जा रही हूँ। जहाँ भी रहो पढ़ाई तो करते रहना। क्या अवंतिका बाई ना, इतना भी डेडिकेटेड नहीं हो रूपाली। तुम्हें गलत फहमी हो गई है। फर्स्ट टाइम आई हो शायद। सर्वजीता मैं जाऊँ? हाँ वो सही कहा एलेक्स। एलेक्स चला गया होगा। क्या कहा लेकिन उसने? देखो तो। नीट फेलियर ग्रेटर देन ब्रेकअप। किस कंटेक्स्ट पे बोला ये? आई डोंट नो, मैंने आधे से ज़्यादा टेक्स्ट स्किप कर दिया शायद। आई थिंक नैन्सी का नीट वाईफाई गया था पहले। ओके। हाई डार्कनेस गेमिंग। चिल गाइस। चिल्ली तो नहीं होएगा ना? आपका तो हो जाएगा क्योंकि आपका एमबीबीएस कंप्लीट हो गया है। हम लोगों का एग्ज़ाम होने वाला है। मुझे ना ऑकवर्ड लगता है सेम लोग ना अगर बार बार मुझे पैसा देते रहते हैं तो। हेलो छोटी बच्ची। आपको मुझे बार बार पैसा नहीं देना चाहिए। ओके नाउ इज नॉट द राइट टाइम। ओके। क्या हुआ? क्या ऑकवर्ड लग रहा है? कि मुझे कोई ना एक बार बार सुपर चैट कर देते हैं, लाइक सेम पर्सन अगेन एंड अगेन। आई थिंक इसीलिए मैंने कल भी एक इंसान को ब्लॉक किया। नाम तो नहीं पता लेकिन आई थिंक जेरस समथिंग कुछ था। ही वॉज पेइंग मी अगेन एंड अगेन, तो मुझे ऑकवर्ड लगा इसलिए मैंने उन्हें ब्लॉक कर दिया, हाइड कर दिया चैनल से। पता नहीं लेकिन ऑकवर्ड लगा। एनीवेज थैंक यू। हाँ लेकिन डोंट पे मी बार बार। सेकंड टाइम भी तो बहुत होता है। मुझे खुश होना चाहिए ना वैसे मुझे जब सुपर चैट मिलता है तो, लेकिन मैं ऑकवर्ड फील करती हूँ पता नहीं क्यों। बिकॉज आई नो इट्स हार्ड टू अर्न मनी, इसलिए शायद। रीड इज अ रूम मतलब? Okay, but you are not forcing anyone. People are super chatting with their own wish. है ना? हाँ इसलिए तो मैंने बोला थैंक यू सो मच। क्या हुआ हिमा? तबीयत सही नहीं है क्या? क्या थक गई हो? नहीं मच्छर बहुत काट रहे हैं तो मैंने ना इसको यही जला दिया, यहीं पर रख दिया, तो शायद इससे ना मुझे इरिटेशन हो रही है। अब मैं बोलना ही भूल गई। आई थिंक ना सर्वजीता ने बताया कि उसका फेवरेट कलर रेड है। वैभव, तो अब तुम्हें पता चला ना उसकी फेवरेट कलर कौन सी है? I'm not that motivated. Take care of your health as well. Study is important, but health is super important. Yep. By the way, what's your name? Smile, please. हाँ रेड। तुम लोगों ने एक स्लॉट कंप्लीट करके आए हो ना, मैंने कंप्लीट किया ही नहीं है। टेस्ट देने चाहिए गई थी ना डार्विन पे, तो आज फिजिक्स का था, फिर लौट के आ गई। मुझे तो अच्छा लगता है आजकल के बच्चों से बात करके। माय नेम इज सचिन। हो, तो आप आजकल के बच्चे नहीं हैं? एग्जैक्टली, आई डोंट लाइक प्लेइंग क्यूपिड, बट मैं हो जाती हूँ, क्यूपिड प्ले करने लग जाती हूँ। वैभव का मुंह तोड़ जवाब दिया, कोई जोड़ी नहीं बनेगी बोल के। गौतम, वैभव, वैभव रोना मत हाँ। तुम्हें कोई अच्छी खासी वो मिलेगी, क्या बोलते हैं उसे, सीए। नेक्स्ट लॉट कब, इफ आई मे आस्क? अभी कर सकते हैं, मैंने ना कब से ब्रेक ही लेके रखा है। अगर आप चाहते हो तो मैं स्टार्ट कर देती हूँ, या फिर मैं चली जाऊँ। वैसे भी मैं ध्यान से नहीं पढ़ रही हूँ ना, इसलिए। वैसे मैंने चाय पी लिया है तो शायद नींद भी ना आए। मैं नहीं पढ़ रही हूँ। मेरी जेई में 55 मार्क्स आए, उसके बाद आई 1 ईयर कोचिंग की, फिर 13 मार्क्स आए। डिक्रीज क्यों हुआ मार्क्स? फिर उसके बाद आपने क्या किया? Then let's do it. Okay. ओके, डॉक्टर डॉक्टर जी बोल रहे हैं कि पढ़ाई करनी चाहिए। वन 20 से मुझे इस बार ना पढ़ने का दिल नहीं कर रहा है, क्योंकि मुझे पता है कि मेरा सिलेक्शन नहीं होगा। जब तुम्हें किसी चीज़ के बारे में आगे से पता है तो तुम वो करने के लिए इतना मोटिवेटेड नहीं रहोगे। शायद, शायद। है ना रचिता, कितना मुश्किल होता है। 25 मिनट का सेशन रखते हो ओके। निशांत, आर यू देयर? तुम्हें नाम बोलने की आदत हो गई है ना, मैं बार बार तुम्हारा नाम लेती रहती हूँ इसलिए। अच्छा है ना, तो उन 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 तुम्हारी मम्मी को तुम पे कोई एक्सपेक्टेशन तो नहीं है, तो तुम थोड़ा फ्री फील करोगी। अम्मा तो क्या बोल रही थी तुम्हें पता है? उस दिन मैंने वो सच्ची में बोला था अम्मा ने, इस बार तो 720 में 720 लाएगी ना, ऐसा करके बोला। इतना खराब लगा। एंड अभी भी उन्हें नहीं पता है कि मैंने एग्ज़ाम में पूरा सिलेबस कंप्लीट नहीं किया है। वो ऐसा बोल रही थी कि अब मैंने उन्हें बोला ना अम्मा वो काला वाला बिल्ली कब ला के दोगी, तो उन्होंने बोला कि वो बिल्ली जब बड़ी हो जाएगी ना तब तक तो तू पढ़ने के लिए दिल्ली जा चुकी होगी ना। उन्हें पता है ना मेरा एक फेवरेट कॉलेज है दिल्ली में, तो उन्हें ऐसा बोला। मुझे इतना ख़राब लगा, गिल्टी भी फील हुआ। तो अगर गाली खा रही हो तो अच्छी बात है, कोई एक्सपेक्ट तो नहीं कर रहा एटलीस्ट। एग्जैक्टली, हम स्ट्रगलिंग सो हार्ड है ना बाकी के कंपैरिजन में, मुझे भी लगता है ऐसा। एनीवेज चलो स्टार्ट करते हैं। ओके, गुड लक एवरीवन। ओके, टिल 2 पढ़ना है तो मुझे सेट करना होगा टाइमर 40 मिनट का। एक्सक्यूज मी। यू आर नॉट गोइंग टू स्टडी विद अस वैभव? 1:30 जितना तो हो ही गया है। गुड लक सर्वचिता, गुड लक निशान, गुड लक श्री राम, गुड लक शुभम, गुड लक निधि, गुड लक श्री, गुड लक अयान, गुड लक वैभव, गुड लक फाइनेंशियल समथिंग एनालिस्ट। तुम तो वंस इन अ ब्लू मून ही शकल दिखाते भी हो, है ना? आई थिंक दिन में भी नहीं थे। Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical. As the heterozygote, one of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's make an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Let us assume as is more common than the normal allele produce the normal enzyme. Let us assume that the normal allele produce the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal less efficient enzyme, non-functional enzyme, and a no enzyme at all, a non-functional enzyme or no enzyme at all. आई थिंक मैं पॉज हो गई हूँ तो यहाँ पर। In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype trait that is a result in the transformation of substrate S. Such equivalent allele pair are very common. But if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Codominance. Till now, we were discussing crosses where the F1 resemble either of the two parents' dominance or was in between, incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different type of red blood cell that determine ABO. A good example is different type of red blood cell, different type of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles, IA, IB and small i. The allele IA and IB produce a slightly different form of sugar, slightly different form of sugar, different form of sugar, slightly different form of sugar, slightly different form of sugar, while allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene allele. IE and IB are completely dominant over I. In other words, when IA and I are present, only IA expresses, because I does not produce any sugar. And when IB and I are present, IB expresses. But when IA and IB are present together, they both express their own type of sugar. This is because of codominance. This is because of codominance. भाई साहब इतना मुश्किल क्यों होता है? This is because of codominance. Hence, red blood cells have both A and B type of sugar. Since there are three different alleles, there are six different combinations of the three alleles. Okay, six different combination of these three alleles that are possible, and therefore a total of six different genotype. And of the human ABO blood group. How many phenotypes are possible? 1, 2, 3, 4, 4. A, B, AB and O. Okay, four phenotype. Genotype of offspring. Okay, parent A, parent B. Allele from parent 1, allele from parent 2. IA, genotype of offspring IA. Blood type of offspring A, IE, IB, EB. B, O. Okay, this is codominance. Nice. Do you realize that example of ABO blood group also provides a good example of multiple allele? मुझे ऐसा लग रहा है कि मैं सच में पॉज हो रही हूँ. छोड़ो कोई बात नहीं, नेक्स्ट ब्रेक में फिक्स करेंगे. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is, three alleles. governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. Do you realize the example of ABO blood grouping? Also, okay, do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than three alleles governing the same character. Since in an individual two alleles can be present, multiple alleles can be found in population studies. When population studies are made, okay, multiple alleles. ABO blood grouping, good example. Also provides a good example of multiple alleles. Here you can see that there are more than two, that is three alleles governing the same character. Since in an individual only two alleles can be present, may produce more than one effect. For example, starch synthesis. Okay, a single gene product may produce more than one effect. That is, ये क्या है? मल्टीपल एलिलिज्म का एग्जाम्पल है क्या? मुझे लगा ये उसका है, प्लियोट्रॉपी का. Dominance is not absolute. Exception of law of the dominance. Okay, multiple allelism. तो ये भी मल्टीपल एलिलिज्म का ही है, नहीं प्लियोट्रोपी का है. तो ये पता नहीं क्या क्या आ रहा है यहाँ. प्लियोट्रोपी का एग्जाम्पल दिया है, तो प्लियोट्रोपी से अलग से ही क्यों दिया है यहाँ पर? ओके. Occasionally, a single gene product may produce more than one effect. Occasionally, a single gene product may produce more than one effect. Single gene produce more than one effect. A single gene produce more than one effect. Okay. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by BB, capital B, homozygotes, and therefore large starch grains. क्यों मच्छर इतना ज़्यादा काट रहा है? मच्छर के बच्चे. मैंने इसके अंदर ही डाल दिया. Here you can see that there are more than two, that is three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles मच्छर के बच्चे को ना जान से मार देना चाहती हूँ. ऐसा नहीं बोलना चाहिए. बहुत ज़्यादा मच्छर आ गया सच्ची में. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by capital B, homozygotes, and therefore large starch grains are produced. In contrast, the small b homozygotes have a lesser efficiency. in starch synthesis and produce smaller starch grains. After maturation of the seed, BB seeds are round and BB seeds are wrinkled. Heterozygote produce round seed and capital B seems to be dominant allele, but the starch grain produced are of intermediate size in capital B small b. So if starch grain size is considered as the phenotype, then from this angle the allele showing incomplete dominance. Okay, allele showing incomplete dominance. Starch grain को size ला एरिया नहीं क्यों सा incomplete dominance होला. Therefore, dominance is not an autonomous feature. Absolute बोल दिया मैंने. Autonomous मतलब absolute है क्या? Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. नींद भी आ रही है अभी तक. फिर से मैं पॉज हो गई. पता नहीं क्या हो रहा है मुझे. मैं पॉज हो रही हूँ बार बार. बाद में देख लेंगे इसको. गया ये भी. Dominance is not autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this products as it does on the particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. Round yellow, wrinkled green. Round yellow. बीज parent को generation के चाहिँ एउटा round yellow लेको छ, एउटा के लेको छ wrinkled green लेको छ. Okay, ry small r र small y. Round yellow. F1 generation सेल्फिङ गर्दाखेरि चारवटा ग्यामेट निकाल्यो. ry capital R small y. Small r capital Y, small r small y. These are the gamete. Okay. Round yellow, round green, wrinkled yellow, wrinkled green. Round yellow, round green. Round yellow, round green. Wrinkled yellow, wrinkled green. 9:3:3:1. 9:3:3:1. इसको तो एक ये भी सही ना. Multiple alleles. Round and wrinkle. WRINKLE wrinkle. yellow and green. Okay, it's 3:1. Again here 3:1. Okay, 3 3 is 9. Round yellow 9 is to round green 3. Okay, wrinkle yellow 3, wrinkle green 1. 9:3:3:1. मच्छर मार दूंगी मैं इसको, मरता ही नहीं है लेकिन बस काट के भाग जाता है। -3:3:1 phenotypic ratio. Okay, genotypic ratio is तुमचा 1:2:2:1:4:2:1:2:1. This is genotypic ratio. This is phenotypic ratio. Mendel also. हाँ मुझे भी लग ही रहा है आज काफ़ी ज़्यादा लैग हो रहा है, लेकिन मैं क्या ही कर सकती हूँ। बहुत सारे मच्छर आ गए हैं ना, तो देखो ना यहाँ पर उड़ रहा है, मुझे काट भी रहा है। आई थिंक मेरा मस्कीटो मीट निकालूंगी मैं। मैंने एक मस्कीटो को पकड़ लिया ऐसा मुझे लग रहा है। आई मे बी रॉन्ग। मैंने पकड़ लिया ऐसा अच्छे से। सी मैंने एक मच्छर को पकड़ लिया। सी मैंने एक मच्छर को पकड़ लिया है।